לפאולה ולשוש. אני מתכבד להזמין את פרופסור מורגן, מנהל המכון לטיפול ומחקר במיאלומה באוניברסיטת ארקנסאו. פרופסור סולומון כבר נתן לנו סקירה על ה-CV שלו, לכן אני לא אחזור על זה. אז בבקשה, פרופסור.
So it's a great pleasure to be here, and I hope I can live up to the honor of being invited. And it's a great pleasure to be here in Israel with you on the timing of your 10th birthday. I've been on the board of the UK patient organisation since it started, and I think patient organisations do a terrific job, and I think it's not unfair to say that patients have become the driving force of research and of advances in clinical medicine for a lot of diseases, but myeloma and sets an example to other diseases. So I'm very pleased to be here to help. I'd be even more pleased if I could make the computer work. Yeah, I'm going to, oh no, here we go, maybe this will do it. I'm going to need help, I'm afraid. Anyway, the title of my talk is Making Cure a Reality for Myeloma Patients. Until recently, I've been living in London and worked at the Royal Marsden Hospital. About 9 months ago, 10 months ago, moved to America, to the University of Arkansas, where we have one of the biggest myeloma practices in the world. And I was asked to talk about a European perspective on myeloma, which suddenly becomes slightly differently when you're living and working in America. Maybe I've cracked it. Okay, now. I'll breathe a sigh of relief. There we go. This is the kind of French view of Europe, of which we're all a part of. You note that Turkey down here is definitely not European, but Israel doesn't really appear on the map at all, and we love the French so dearly. But this is my normal environment. I'm actually a child from the west of Britain, from Wales, the place that's highlighted in red there. But I've recently been transplanted to a new microenvironment, this time in Arkansas. And for most of you who have no clue where Arkansas is, much like myself until a few months ago, that is exactly where Arkansas is. Fortunately for me, there's a London, Arkansas, so I've not really moved all that far from home. Of course, it's a different world, and you, like myeloma cells, you have to adapt to your microenvironment, and storms like this in America are really common and kind of worrisome. But in Wales, this is simply what we refer to as summer. And again, this was me as a child. I was always looking forward to summer, not because of the sunshine, but simply because the rain became warmer. But this is my new environment next to the Arkansas River, and Faith and I live on the hill just above this bridge. But cutting to the important messages, though, this is the enemy in this fight. These are myeloma cells that live in the bone marrow. This is what a cell looks like. That's the nucleus. and there is the nucleolus. And this is the brain of the creature that lives in people's bone marrows and causes such destruction. And what we really try to do as clinicians and scientists is to understand why these cells develop, but really the purpose of that understanding is how do we kill these cells so as we can restore people's lives to normal. And this tries to summarize what it is we're trying to do. So if this is that myeloma cell with its brain here, so this brain is sustained by signals from outside the cell, which give it a survival advantage, which allow it to live. So if we could understand those signals and design treatments that switched off those signals, we can induce this cell to undergo suicide and to commit what we call apoptosis. And so that's the purpose that underlies much of the science that goes on. I'm hoping to communicate a number of messages to you today. I can never be sure that I can communicate even with other scientists. So I'm going to try and make a distinct attempt to communicate with you as patients. So a good way of doing that is to just outline what I'm trying to say, so when I'm stumbling through it, you actually understand. So important message number one, myeloma is not a single disease. It's a collection of diseases which look the same. This is very important because you should treat each of the diseases in a specialized way designed for that disease. About 15% of myeloma is high risk. High risk disease is bad. High risk disease represents a serious challenge for clinicians and scientists to recognize it and to treat it appropriately. In contrast, 85% of myeloma is low risk. And for low risk myeloma patients, cure is definitely a reality. There is no doubt about that we're bringing cures to more and more people, but it's predominantly within the low risk group. This has been a good year for myeloma patients. It's never a good year to get myeloma, but this year there have been significant progress, and by the end of the year, there'll be 3 new treatments licensed for myeloma patients that I think will be game changers and will bring more cures to more people. I'd like to make the point, as I said earlier, that we should develop treatments going forward, not for myeloma in general, but for subgroups of myeloma, because by splitting the disease up, we will make more progress overall. And the final point is that because high risk remains a challenge, new trials and strategies are needed for that subgroup. At Little Rock, we've been pioneers about aiming for cures in myeloma. There are 2 ideas that are out there. One is turning myeloma into a chronic disease, which I'm very approving of, but I'm more approving of the idea that we should strive to cure myeloma completely so that patients are off treatment for many, many years and live as long as other people in the population. Why would we not strive for that as a goal? If a side effect of that was we turned myeloma into a chronic disease, what would be wrong with that? Despite me saying there are cures, I think it's very important that you get the best treatment if you are going to be cured. And I think one of the key features in that is to work with your local doctor because they're close to you, they give you emotional support, they can make things available for you. But why wouldn't you ask for a second opinion and get an expert opinion from a myeloma specialist? It's even difficult for me to keep up with the literature, so you wouldn't expect a general doctor to be able to do that. So good advice is to always seek an expert opinion. The new treatments that are coming will improve the cure rates for myeloma. And one of the concepts that you have to embrace about cure is that if you're going to cure, you should deliver the treatment aimed for cure when patients are first diagnosed. So we should improve our diagnostics, we should diagnose patients earlier, and we shouldn't wait until patients have crippling bone disease, renal failure, and anemia before we start treatment. So I promised Paula that I wouldn't show lots of survival curves, but I'm going to go back on my word just a little. This is to sort of show to you that there are many different groups of myeloma. In fact, there are 7 different groups that have different survivals. They respond differently to treatment, and it's important to know which group you fall into. Each one of these columns, and there are approximately 400 columns, are an individual patient's gene expression profile from their myeloma cells. So we select the myeloma cells, look at what genes are expressed, and then if you put 600 together, you can cluster everything using a computer algorithm, and it forms these really rather beautiful red subgroups. And these are the different groups, and those are their percentages. And so this I really did promise not to show, but each one of these groups is given a different color, and these are survival curves. And if you're up here, this is a good thing. If you're down here, this is not a good thing. So you can see the different colors have different slopes, arguing that these cases are behaving differently in the clinic and responding differently to treatment. The other thing you can do with this technology is define this 15% of high-risk disease, which is really important. And again, you can see this is, these are survival curves again, but you can see here the high-risk disease does much, much worse than the low-risk disease. So it's important information because it affects the way you should be treated. And this, just for final complexity's sake, shows the survival for these different groups. And you can see that the different names have different slopes. The red curve was where we were 15 years ago, and you can see we've shifted up the curves for these subtypes and made significant progress in the last 10 years. But for these subgroups, the MF subgroup and the PR subgroup, we've made less progress. So what's the message from those complicated slides? Myeloma is not a single disease. You can use diagnostics to split it up into clinically meaningful groups, and each of those groups can and potentially should be treated differently. So moving to this high-risk disease, this is proven very, very difficult to treat. In the last 15 years, we've not made any significant improvement at all, which is deeply in contrast to the low-risk disease where we're curing people. It's only 15% of presenting patients, but it's important to know about it, and it needs this special GEP test to identify it. If you relapse, you can go from low risk to high risk, and that happens about 20% of the time. My thought is that the new antibodies that will be licensed by the end of the year will improve the outcome of this group significantly. The other changes that are really beginning to make a difference are gene expression profiling, which I showed you, this new technology called gene sequencing, where you can look for the genes that are affecting those survival signals, which I showed you in that first slide. Using these techniques, you can pick a treatment for a specific patient. The other important advances are PET scanning and this MRI approach called a DWIB. So it's diffusion-weighted MRI. But the imaging of myeloma, I think, is going to drive progress quicker than people imagine as we start to see low levels of disease before bone disease has developed that are amenable to treatment. So moving to kind of what I think, or why I think 2015 is a good year for myeloma patients. Carfilzomib, a new proteasome inhibitor similar to Velcade, is becoming available generally. Pomalidomide, a new IMiD drug like thalidomide and Revlimid, has become available. Those are being used by patients. This drug called a histone deacetylase inhibitor, an HDAC, and an example is panabinostat. So panabinostat was approved a few months ago and is now available for patients with multiple myeloma. These two new antibodies, which are like magic bullets that recognize the bad cells and go directly to them and kill them, will be available by the end of the year, I suspect. The anti-CD38 antibody, daratumumab, is likely to be available. And elotuzumab, an anti-CS1 antibody, has had good results in studies and hopefully will be licensed by the end of the year. So these are new ways of using the patient's immune system to target their cancer cells and kill them. I would make the point, though, that new drugs can improve on the standard treatments but are not ready to replace them. And I wouldn't move away from stem cell transplantation at this stage for patients who can tolerate it. I think these can improve the outcome of stem cell transplantation. The real aim for the future, which is very much alive and well here in Israel, and we heard from a prostate cancer scientist yesterday and a colon cancer scientist who came to the meeting that was organized by the committee. These are really being developed, and I think your challenge is to introduce this personalized, targeted treatment into the sphere of multiple myeloma. And the concept is to understand the nature of the myeloma cell in a patient, and then from a selection of treatment, to select the best treatment with the fewest side effects that gives the most number of responses and ultimately cures. And so this is where we were with a one-size-fits-all approach. All patients are the same, all treatments the same. But this is where we should be. We should be able to recognize the different components of this jigsaw, pull them out into their individual groups, and to design treatment strategies for these individual components of the jigsaw. And by doing that, using a diagnostic and these targeted treatments, I think we can really make improvements more rapidly even than we have in the past. We've been very successful in pushing progress, but we need to continue making progress. And I think this is the way forward. And so I would thank you for your attention. I'm very happy to answer questions later on during the break, and I hope you have a very productive and successful day.
תודה לפרופסור מרטין גארט. Thank you very much. כפיים עוד פעם הייתי מבקש.